looks like we should get started. Things are good? Hanging in there? Excellent. Yeah, we're missing more people because of exposure reasons, and I don't know what, but I'm glad you all group here just are here. Does anybody like those puzzles? I don't know if you've ever seen them, where like you have a box and like it unfolds and trying to figure out which side when folded together is on the top. Have you seen those puzzles before? Like spatial awareness puzzles? Anybody here like those? Because if you do, today is your day. We're looking at some of the structural parts of DNA, like its shape and how it gets that way. Next week, we're going to look at what DNA does. Today, and again on Friday, we're looking at basically the shape of the stuff. All right? So, you've seen this picture before, right? It's this model here, yeah? It's an elegant molecule. It's not super easy to describe its shape. I mean, the term is double helix, and this part is falling off. I needed some sort of clip thing here. But it's like this, right? We've got these two different strands running together. They're in a position called, is this screen viewable? Is this okay? Okay, I'll turn the lights off later. It's in something called anti-parallel organization, meaning this end here has the 3' end going one direction, and this other strand has the 3' end going the other direction. So like, things that are parallel are lined up with each other. These are lined up, but they're flipped. We're okay with this? If you're actually looking, like, all the drawings are usually flat, but if you're looking for like an actual space-filling model, it's going to look something like this, right? It takes up physical space. It's not like entirely 2D. So, Watson and Crick, based on Franklin's work, figure out that this is basically the shape of it. And at first, when they're looking at it, they're looking at it, they're like, okay, so we think maybe the bases are pairing with like bases. Maybe all the A's are pairing with A's. And that doesn't work, though, because the spacing of the molecule then isn't consistent. It's either too fat or too thin. So, like, okay, like's not pairing with like, so maybe it's just the purines are pairing with other purines. Like the fat ones are pairing with fat ones, the small ones, the pyrimidines, are pairing with themselves. That doesn't work either, because then the shape's like lumpy. It's not consistent. But if you pair a purine with a pyrimidine, you've got a consistent width. And this works beautifully. It's actually more specific than just purine pyrimidine. They pair in a very specific pattern. So adenine always pairs with thymine in DNA, and guanine always pairs with cytosine, and only pairs with that in DNA. We're okay with this idea? And this actually explains something that an earlier scientist called Chargaff had figured out. He figured out that in whatever organism you're in slime mold, in sponges, in broccoli, in people, whatever organism you're in, if you know the amount of the A nucleotides, you know the amount of the T nucleotides, right? Help me out with how you can do that. So, if I know the amount, if I know the percentage of A's, do I know the percentage of T's? Let's count really quickly. The A's are the red ones. So we have 1, 2, 3, 4, 5, 6. Is that fair? many T's are in this? Do I need to count those? Nope. Fair enough? Because they're around the same amount, right? Help me out with the whole just like quick, brief algebra. Let's say I have 10% A's in my whole thing. What percent of T's do I have? 10% as well. Fair enough? Okay, so I know 10% A, I know I have 10% T. Does that mean I can now know what percent C I have? How? So if I've got 100% total, I've got 10% A, 10% T. So there's 80% left. How much of that should be C? 40. How come? You're totally right. Because it has to pair. Do we see how that works? So if you know the percentage of any one of the nucleotides, you can know the percentage of all the rest. Because they have to pair and have to pair in specific order. Do we see that as a concept? And it had, when Chargaff had figured this out, it had kind of been just like an oddity, kind of like that's interesting. But the structure is the reason for this. The structure is because they're only pairing in this pattern. That explains why you can figure out the ratios so well. So yeah, these bases are pairing in a specific and known order. This is cool. This sets up DNA replication and repair. Now, I got excited with the 9 o'clock class and launched into this whole discussion of like information transfer, which I'm now wondering if I took too long doing that. Briefly, super briefly, did any of you all at some point in your life talk about like the Egyptians? You learned about like hieroglyphics at some point in your life? Yeah? Okay. It's kind of cool, right? Did you write your name in hieroglyphics? You're like, I identify strongly with the pyramid shape or whatever. You're like, this is me. Okay. Pictogram writing is cool. It's great. It works well for many things. You don't have to sound anything out. You just have like, your picture of a pyramid and you're like, it's a pyramid. Right? You can get more complex than that. You have like concepts and ideas, right? But for every single concept and idea, you need a picture for it. Right? This becomes problematic. Right? Like if you're trying to type and you just have every single key be a different picture concept, it becomes hard to get that done. Right? With using letters, you can have a very small amount of letters. You can keep combining in different ways to mean all the different concepts, right? DNA is going to try to do that similar ideas. It has a very, very few number of options. It has a very few number of A's, G's, T's, C's. But it's going to put those together in interesting combinations to represent the concepts of all these proteins. Fair enough? But we're using DNA because we're wanting to pass on this information for how to make all these different kinds of proteins. DNA replication happens when you're going to undergo meiosis, when you're going to, say, make gametes, right? Is there any other time that you personally undergo DNA replication? Yeah. Yeah, so like let's say you scrape yourself, you suddenly need some new cells. Yeah, your cells are absolutely going to divide, and your original cell has DNA. The new cell better have DNA too, right? Is everyone okay with this? If you sit there quietly, you can just feel yourself replicating DNA, right? I mean, you can't, but like, you are replicating DNA. All over the place, your cells should be doing this, right? It happens in meiosis, yes, a little bit. It's happening all the time in mitosis, okay? Just you're replicating DNA all over the place. Is everyone okay with this? It's not like a weird thing for the body to do. Cells do this. And DNA is a really good model, is a good molecule to get this done because, as you all have cleverly figured out, if I know what this strand is, I know it's T, A, C, T. I know, without even having to look at this, I know that this other strand is A, T, G, A. If I know one strand, I know the other strand. So, in DNA replication, the parent molecule unwinds. And the parent molecule is not the, like, reproductive, your actual mother and father. It's the original DNA strand we're talking about in a given cell. Okay? It's called the parent strand because it's the original one. It's going to unwind, and then you're going to make two new daughter strands based on the rules. So it's this picture here. I'm going to keep showing DNA flat because it's tricky to show it curved, but we're all okay that that picture is just this, but unwound, like flattenedified. Yeah, okay. It's a word, not really, but. So you pull it apart, you separate the parental strands, and then you make new, they're called complementary strands, to the template strands. The template strands, the original, you pull apart, you now have two template strands, you're going to make two new daughter strands, and you went from having one piece of double-stranded DNA to now having two pieces of double-stranded DNA. Everyone's cool with this concept? Seen it? Pretty straightforward? Make sure I'm actually recording. I suddenly had this realization I should check. Yep, doing great. Okay, lots of words here for your personal study, but I'm going to show you this. When people were first looking at DNA replication, there were a couple options for how DNA could get replicated. The three possible models were these ones, okay? So people knew, okay, you're going to go from having one double-stranded copy of DNA to two double-stranded copies of DNA. And there are a couple ways you could do this, theoretically. There's only one way that works. We're going to look at the options. One option is called the conservative model. And that model is like, all right, you have your DNA, and so whatever replication machinery is going to replicate this. We're not worried about how it's happening right now, it's just that it does. Whatever replication machinery comes, looks at this, and basically makes a photocopy of it, more or less. It's called the conservative model. It's up here. It's saying this dark blue one is the parent model. And it's saying in a conservative model, this strand is entirely conserved. Nothing changes from here. The same physical atoms that are in this one are in here. And there's a copy of it up here. And then when this one replicates again, you'd have this one stay the same, but you'd have a copy made of it. This one stays the same, but you have a copy made of it. Do you see that version? That's the conservative replication model. The semi-conservative replication model, I really hope you like this one, because this is the one that works, but like, just look at here, is, oh, the lighting's bad. Is that better? Yeah. The semi-conservative one is, all right, you have this, one original strand stays entirely intact, and shows up here, and you make a new strand to go along with it. One strand stays entirely intact to make a new strand go along with it. And if we're following this strand here, if you pull apart, this strand's up here, this strand's down here, and there are two new strands. Same with here. The final idea that was a contender model, though it's a mess, is what's called the dispersive model, which is where it's like, yep, you keep some involved, but you kind of just chunk it up. And so little bits of the original one are spread throughout everything. Okay? The experiment that was done to check this is elegant, so we're going to look at it, because it's just so beautiful. It's by Messelson and Stoll. What they're doing is this. They're growing up bacteria. Sorry, I feel like I need to show you these experiments because they're like the basis of some very important biological concepts. And they're not that complicated. They're clever and they're elegant, but they're not like superly hard to do. Which honestly is the basis of a lot of good science, is just a clever way to figure something out. So what they've got, let's throw my DNA models around. They've got bacteria that they're going to grow up in a heavy version of nitrogen. So nitrogen shows up in DNA. You can have different isotopes of nitrogen, so the atom behaves the same way, except it's a little bit heavier than regular nitrogen. Okay, are we okay with this? So then you can find it again if you're looking for it. So you grow up bacteria in heavy nitrogen, and it has other stuff too, but the nitrogen's heavy, so any DNA that's grown during this normal time is going to be slightly heavier than normal. You then take your little bacteria buddies, and you throw them in with new nitrogen. And so now, when this bacteria divides, and it looks to build new DNA, the only DNA it can build is going to have lighter nitrogen in it. Is everyone with me here? After it's replicated one time, they took this out, centrifuged it down, and looked to see where that band of DNA was. Right? So centrifuge, you've got stuff, you spin it, denser things go hit the bottom, less dense things stay up top. And like this shouldn't surprise you, right? If you had, you've got a tube, it has jello in it, and you put a rock and a leaf both at the top, you spin around for a while holding this. After you've done this, the rock's to the far end, and the leaf still is near the top, just because of their densities. You okay with this as a concept? Okay. So, first time they do this, you've got this band here. You wait till it's divided a second time, and do the same thing, and you end up with two bands. You end up with a band that's right here, where this one was, and you end up with a band that's up here. It might not be intuitively inherently obvious that this proves that it's semi-conservative, but let's look and see what would have happened with other things. Is everyone okay with what's going on in this picture? Like how we have this? The take-home message is something like this. If you had the conservative model working, right, like if the model where one DNA strand stays entirely intact and just gets photocopied, then you would have had, back here, you would have had, this stuff would have been really heavy, right? You throw it in here. The heavy one would have stayed. There would have been a photocopy of a lighter version, and you'd have had a heavy band and a light band of DNA. Do you see that? But since in the first replication you don't have two bands, you just have this one band, it can't be the conservative model. Do you see that? Like that doesn't work. If it had been the dispersive model, where things are just like all over the place, in the first version, yeah, you'd have seen a band right here. But in the second version, you wouldn't have two bands because everything's getting mixed up and you'd basically average it out. So you'd end up, instead of this band, you'd end up with a band like here-ish, instead of two different bands. Do we see that okay? So this experiment showed that, like, yep, it's semi-conservative. Like, this is how it's working. Questions, concerns? We're all on the semi-conservative model. The rest of today, we're looking at how this actually goes down. It's called DNA replication. It's copying DNA. DNA is really good at getting copied. It's fast, and it's accurate. And there are lots of things that need to happen. Replication is going to start what's called an origin of replication. It's where the two DNA strands start to get opened up and they make a bubble. It's legitimately called a bubble. Bacterial DNA is circular. It doesn't have like ends. It's just like a closed loop. And so a bubble will start, it'll keep opening up, opening up, opening up, and you'll end up with 2 double-stranded DNA bits at the end once you've replicated it. Are we seeing how that's working? You can see this right here is the replication bubble. It's being pulled open and DNA is being built on both sides. Yeah? Okay with that? In bacteria, you're expecting one replication bubble that's going both directions, opening up fast, right? In eukaryotic cells, you're actually expecting a lot of replication bubbles to be opened at the same time. Eukaryotic DNA is going to be linear. It doesn't loop back on itself. Like it twists off, but it's not connected at the ends. You can see it here, right? You've got a replication bubble here, another one here, a really big one there. They eventually run into each other, right? So like, as this one opens up and that one opens up, they'll bump into each other, and that's great. And then this actually has been probably a couple that bumped into each other. But it just keeps going until you've pulled the whole bit, replicated the whole bit, and you now have 2 daughter copies of DNA. Is everyone okay with this? A lot of this, and it gets more so, a lot of this, like when you see it, you're like, huh, that's elegant. The trick is remembering it later. But if everyone's feeling comfortable with it now, that's great. I have a video for you people. I'm going to try, I don't know if this is going to work, I'm going to try really hard to click the button to play both the computer version for the online people and this version at the same time. I'm not sure if this is going to make it terrible or beautiful, because I'm not sure if I'm coordinated enough to do this at the exact same time. We're going to try. Online people, if it doesn't work, just watch it on your notes. Come on. And the problem is the
Replication of a DNA molecule begins at a specific sequence called an origin of replication.
Okay.
The 2 strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the 2 daughter molecules consists of 1 old strand, shown in purple, and 1 new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in 2 daughter DNA molecules.
Everyone's cool with this? Alright. We're going to look at this picture here. There's a lot going on. None of this stuff here is actually copying DNA yet. We're just going to get DNA set up to be copied. So a couple things to know the terms. The part where it opens up, this is called the replication fork. Okay, because it's like a fork in the road, it goes 2 ways. Replication fork right here. You know, like when you have a zipper on your shirt, there's like the actual zipper part, but then there's the little doohickey that like pulls up and down. Does anybody know the name for that thing? Because I legitimately do not. Okay, but the zipper doohickey, which I call a zipper, but there's also like the physical zipper part, that you can use to like unzip the zipper. Yes? Helicase does that. This is a helix. Well, it's a double helix. This is a helix. A helicase is an enzyme that undoes it, that pulls it apart. It's unzipping DNA like a zipper unzips a zipper, which I need a new word for the zipper part. Are we okay with that? The helicase is going to move along and pull these 2 strands apart. Concerns, questions, we're fine. It's not copying anything. It's just opening this up so we can get it done. Right? We need that to happen. Has anybody ever used packing tape and you've got a long strip of packing tape and you now need to do something with it? What's the challenge that arises with a long strip of packing tape, especially the really sticky kind? What happens if you're not being uber careful? It gets stuck to itself. Have you had this happen, like, sticks and then you're trying to, like, pull apart in this mess, you're like, ah, my life is terrible? Yeah? Okay. Maybe you shouldn't go, my life is terrible if this tape is sticky. But, like, it's a struggle. DNA sticks to itself, right? DNA, the A's stick to the T's, the G's stick to the C's. Once you've opened this up, DNA wants to do two things. Thing number one, it wants to stick back to the other strand. It wants to come back down because it was like conveniently stuck together a minute ago. You pulled it apart, it's still like sticking to itself. It would want to stick back together. It also, so that's a problem. It can also stick to itself in weird ways. Like let's say you have a whole bunch of A's right here and a whole bunch of T's right here. They could fold back over and stick to itself. You don't want that. You've spent a lot of effort getting helicase to pull this open. You don't want it to just collapse back down or to stick to itself in weird ways. No. Bad. Right? So you need something to stabilize this. So you're going to use what are called single-strand binding proteins, which are basically going to make it so that this DNA can't stick back itself. Okay? You're going to basically just hold it, you're going to stabilize it. Fair enough? So we've now unwound it and stabilized it. Two more things we need to worry about before we can get to copying it. Has anybody in here ever had, like, long hair? We're talking, like, to the waist long hair. Anybody? Yeah. Can you tell us what happens as you've got long hair, you're starting to braid it at the top? What happens to the bottom? Does it braid itself back up? Do you know what I'm talking about? Like you're braiding and twisting one part and then the other part starts doing it at the bottom? Yeah, and it's frustrating because that's not what you want to have happen? Okay. This thing, and for many reasons, including like the death and despair reasons, I dislike the pandemic. I also dislike that I can't have people come up here and like do interactive things with like models and ropes and who knows what. But I'm gonna just try just with my one little model here instead of you guys doing stuff. It's very sad. You got this DNA. As you pull this apart, the same number of twists that are in it are in it even as you pull it apart. But you just push the twists to the edge. I feel like this has happened to me as I've done things before. I'll use a bad example. If you've got anything that's twisted, hi online people. You've got something that has a twist in it, right? If you open up an edge, right, you don't actually untwist anything. All you're doing is jamming the twist to one side. And this is going to get tight. It's going to get tight fast. And it's going to become a problem. DNA is going to have a similar problem, right? This thing's coiled. As you push that helicase along, you're just cramming the coil further down. Do you see that as a problem? It's going to make it get tight. It's not going to be able to do its job. So, that thing that's called topoisomerase is going to solve the problem. It's this enzyme that its job, it's not unwinding, it's not opening up DNA, it's not replicating DNA, it's not stabilizing DNA. It's going to just correct the overwinding. So it goes ahead of the replication fork, and it basically breaks the DNA, the double strand there, it lets it undo its overwinding, and then puts it back together. Does that make sense? It's basically taking the pressure off the coil. Is everyone okay with this? So we have all these things going on just to get this ready to go. One last thing, the enzyme that's actually building DNA, it's called DNA polymerase, it needs an end to stick to. It can't just attach, even though it wants to have a matching side to like whatever given nucleotide, it can't just stick it on. It has to have a free 3' end to add onto. So you're going to need, before you even start replicating, you need something called a primer. It's made out of RNA, and an enzyme called primase is going to add a little RNA primer, which is going to give you a free 3' end that you can then add DNA to. So far, so good with this? A lot going on. All of this is just to set up so that DNA can get replicated. So this primer that gets added is short. It's like 5 to 10 little nucleotides long. It's just going to be the starting point for the new DNA strand. When you do this, the enzyme that's doing this is called DNA polymerase. It's going to make a polymer of DNA, a long polymer of DNA. That's its job. Bacteria are crazy fast at this. It's going to grow at about 500 nucleotides per second. This DNA polymerase in bacteria is just crazy fast. Boom, 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 puts it on, right? Has anybody here ever tried to type really, really quickly? Like super speed, right? Your fingers are flying. Yeah? Right before an essay is due? No? Okay, you people, the kind of responsible people who have never looked at the deadline and be like, it's due in an hour, I should start now. Go you, congratulations on not being procrastinators. If you can imagine procrastinating until way too late, remember your paper by the way, people start typing faster. They typically make more mistakes the faster they are, just because, they're going fast. Bacteria DNA polymerase is really fast. It's not as accurate as yours. Yours is way slower. It's still really fast. It's DNA polymerase is going about 50 nucleotides per second, which isn't bad, and especially if you have lots of origins of replication, you can get done in no time at all. But it's not as fast. But you don't make as many mistakes. So that's it. Bacteria don't really care if they make some mistakes. Their goal is just to have a whole bunch of them. Your cells really care about not giving you cancer, amongst other things. Lots of words to say this picture. Each nucleotide that gets added to a growing DNA strand is something that's called a nucleoside triphosphate, which is a fancy way of saying it's this little nucleotide buddy. It's floating free, and it's got 3 phosphates on it. You okay with this? You've actually seen adenine triphosphate. You saw ATP? Yeah. You've seen the A before. The other kinds are going to be very similar, except just their little nitrogenous base is different. So, honestly, ATP has ribose on it. This kind of stuff for DNA has got deoxyribose. The sugar's a little bit different. This, when it came on, was basically ATP, except the sugar had one less oxygen. But okay, this little nucleotide's going to come on. DNA polymerase is going to read the template strand. So it reads this template strand and says, that's an A. If that's an A, I need to add a T. Okay, that? If it saw a G, what would it feel like it needed to add? A C. Okay? It reads the template strand and it's going to add the complementary base. Okay, it's the base that matches not because it's the same, but it matches because it's the version that fits. It's going to read the template strand, but it's going to add that nucleotide to the growing chain. So it's going to add it to the growing strand. Fair enough? Are we okay with this? It can only attach to a free 3' end, this end that has the oxygen on it, this OH group on it. It cannot add to the end that has the phosphorus bit on it. Fair enough? Are we okay with this? It can only add, so as the template strand is being built, it can only, as the template strand is being read, it can only add the daughter strand this direction. Is everyone cool with this? And it's just going to keep going. You're going to get rid of this phosphorus bit, and you're going to keep just growing this chain along. So far, so good? So, yes, terms. Because DNA is anti-parallel, one strand's 5' to 3', the other is 3', 5'. Replication happens in some funky ways. So, because of how this is organized, the growing strand has to grow from the 5' end to the 3' end because it's adding on to the 3' end. Do you see that? The prime is just a numbering system. It's a numbering system where you're counting carbons, and you're saying, this is carbon number 3 end. So you're only adding to that side. We're spending the rest of today, we've got time, looking at this. We've got this replication bubble that's opening up. Do you see up there? Okay, it's opening up. Let me actually show you the video of it, and then we'll talk about it. I'm going to try it again. Last time was actually okay.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The two strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
We're okay with that picture? Great. Instead of just staying on that, I'm gonna yap about it, even though I realize you're sold on it. We've got this thing going on here. We're zoomed in up on the replication bubble, and we're actually zoomed in up, and we're just looking at this part right here. The helicase is wandering along, opening this up. Topoisomerase is fixing the overwinding ahead of it. We put an RNA primer on. The enzyme, cleverly called primase, put the primer on. And then we've got DNA polymerase. It has the 3' end from the primer that it sees. It can put new nucleotides on, and it's just going to keep going. So long as this helicase keeps opening up in front of it, it can just keep running along. Fair enough? This is called the leading strand, and it's just going to keep chugging along. It doesn't have to stop or start. It can just build for forever. And you just saw this. Now, there's the other side of this replication fork. Along one template strand of the DNA polymerase has to jump. It's called a lagging strand. I'm actually going to show you the video first, and then we're going to look at the words, pictures. Aha. Here's this animation, and then we're going to talk about it.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand.
Okay, does that picture make sense when we're watching it? It's okay? That's this all here. Okay, this is everything shown together in one picture. We saw, a while ago, we saw this leading strand that was up here, that so long as helicase was opening enough, it could just keep going along. Because it has a free end, just add forever, right? Or until you run to the next bubble or the end. The lagging strand has the opposite problem, where as helicase opens this up, you expose this DNA that needs to get replicated. But whereas this one with the primer that had the 3' end, this one's got the 5' end, so it has to keep starting and restopping. So the bubble opens up. You add an RNA primer. You've got primase here. You add this little chunk here. So you have a free 3' end. Great. Then DNA polymerase comes along, builds. It's going to run into this last little bit they just saw. Boom. It's going to have to pop back off. Meanwhile, this has opened up some more, so you can go ahead and put another RNA primer on. Run DNA polymerase on again. It's going to run to this bit, come off. It's going to be built in these little jump sections. Do we see that? Is that okay? It's going to build, run into where it started, have to start back over, build, start back over, build. Yeah? The leading and lagging strands are both being built at the same time. The lagging strand, though, is going to be made in lots of little chunks. And you've got these primers. These primers are made up of RNA, which is great, except you're trying to build DNA. So you need this it's another DNA polymerase that's going to come and replace the RNA with DNA. And this time it works because you have a free 3' end right here that you could put the 3' end, build. It's hard to see here. Let me make it bigger. All the things. There. Between those fragments, there's a break right here. So DNA, well, actually, for a reason, you'll build DNA right here, you'll build DNA right here. There'll be a break there. This enzyme called DNA ligase fuses those together. So you end up, the lagging strand gets put together into one big piece. We're okay with this? Here's the picture of 1/2 of the replication bubble. All right. The other half. So watch them. Look at it right now. You've got this bubble opening up. Topoisomerase is doing its thing. Helicase is doing its thing. It's right there, moving along. The single strand binding proteins aren't shown, but they're doing their thing. Helicase unzips the DNA. DNA polymerase on the leading strand just build. On the lagging strand, adds a primer, DNA polymerase, a primer's added, DNA polymerase, it's going to jump. Another DNA polymerase is going to replace all these little primers with DNA, and then the ligase is going to connect those chunks together. This is on 1/2 the replication bubble. Meanwhile, the other half, because it's going both directions, right? It's being pulled open. 1/2 is being pulled open this way. This other half where it's getting pulled open the other way, there's another helicase on that side. Up here, this is the leading strand going that way. On this side of the replication bubble is the lagging strand. Meanwhile, this part's the lagging strand here, but on the other side is the leading strand. Do we see that okay? One more video to watch. This is trying to show you the whole thing at the same time.
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand, while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Yeah? Okay, actually, so I lied on accident. I have one more video to show you. Sorry, questions about that? Today's class, I like it. It's important concepts, like DNA is important. The structure is important. It's not super exciting because you're like, yep, it's a physical structure. That makes more physical structures. It's important, though. You need it to be fast, you need it to be accurate, and you need it to be able to, do this copying thing. Last video, I think I actually have time. Can we all take a moment to appreciate that we totally got through all the slides? There's the things to think about. And this legitimately is the last slide, and we have 2 minutes, and it's beautiful.
Copying DNA, a process called DNA replication, is very simple. The two complementary In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Great. If you only, this one's a second behind us. Great. So if you only have like brain space for one slide, this is the one to like look at. Questions? We're good? All right. I will see you on Friday. Have a wonderful weekend, or not weekend, Wednesday. Remember your bio lab stuff.